So today we're going to discuss what's called the Central Dogma. And what this means is essentially the flow of genetic information in the cell. So we sometimes call this protein synthesis. So synthesis meaning to produce, and that's what it's really about, is producing a protein product that's instructed for by a gene. And so there are many genes, and you have the same DNA or the same genetic material in all of your cells. However, some of those genes are not being turned on at the moment, right? Some are turned on, and by turned on we mean that set of instructions is being produced in the cell into its product, the protein. So if you think about if you were going to make brownies, you have instructions on the recipe for how to make those brownies. That would be essentially like the gene, it's the instructions. And then you have the eggs and the flour that go together. You mix them up, you follow the instructions, and the brownies are actually the product that's produced. That would be like the protein in the cell. So the gene is what we would call the genotype. Okay, that is the genetic material that's inherited right in the cell. And for our cells, which are eukaryotic cells, that the DNA or the genetic material is going to be located in the nucleus. However, if we're talking about prokaryotic cells, since there are no organelles, there's no nucleus, therefore the genetic material is in the cytoplasm. The protein then is what we call the phenotype, and again, this is the product that is instructed for by the gene. Now, in the Central Dogma, there is what we would call a middleman, mRNA, or the M stands for messenger RNA. And if you look over here to the figure at the right, you notice that it starts with DNA, and I'm going to go ahead, even though we know that not all cells have a nucleus, I'm going to go ahead and designate, at least for our cells, that the DNA is packaged in the nucleus. And then we have this process that turns the instructions in the DNA into or it produces an mRNA message. Okay, so I'm just going to put here, this is the message. And so we're going to put an M in front of that RNA. Then we have another process that actually produces the protein product. Now, if you'll recall, a protein is produced on something called a ribosome. Well, the ribosomes are found either in the cytoplasm or in a eukaryotic cell. They can be found on the rough endoplasmic reticulum. So if the DNA is in the nucleus and the protein is produced on the ribosome, we have to have a middleman to carry that DNA message to the ribosome. So that's how you can keep that straight of why we need a messenger RNA. Now, what specifically do we mean when we're saying a gene? We talk about DNA, we talk about chromosomes, we talk about genetic material, we talk about gene. Let's get it all straight. So, genetic material, right, is everything that's inherited. Okay, so that would be the DNA. Well, the DNA is actually packaged around proteins called histones. We learned about that in the last lesson. And chromosomes are just one form that the DNA can take. So if the DNA is very tightly coiled or wound around those histones, it's in the form of chromosomes. If it becomes less tightly packed, it's called chromatin. Now, a gene is just a short segment or a certain number of nucleotides in a DNA sequence. And again, it would be found in the nucleus if we're talking about eukaryotic cells, or the cytoplasm if we're talking about prokaryotic cells. So in our figure that we looked at before, we said this is one process between DNA to RNA, and that process is called transcription. And if you think about the word transcription, what does that mean? I think about years and years ago, before we had copy machines or computers, and if another copy of something was going to be made, it had to be done by hand. And so let's say that I have a manuscript that someone else had written, and I wanted to make a copy of that. As I'm reading it, I'm transcribing or writing down. Well, it's the same message, and it's in the same language, but my handwriting looks a little different. So there's, small differences there, but for the most part, it's very similar. It's just making another copy of that. That's similar to transcription because we're going from DNA to RNA, and those are both nucleotides. So it's it's the same alphabet for the most part with a slight difference. We'll talk about that in a minute. But it's really a very similar sequence. Now, I'm going to use the word copied, and we'll talk in a minute about what we mean by copy. But for the most part, the DNA message is read and made into an mRNA sequence. And what is doing that is this enzyme called RNA polymerase. Remember, ASC is the last three letters that indicate that's an enzyme. You may recall that we also learned about an enzyme called DNA polymerase when we talked about replication, and that's because the new molecule being produced was a DNA strand. In this case, it's an RNA strand that's being produced. So where's this happening? We talked about this a little bit already. Depends on the type of cell. If it's a eukaryotic cell, transcription then has to be happening in the nucleus where the DNA is. If it's a prokaryotic cell, then it's happening in the cytoplasm. All right. Translation then is this portion here between the mRNA and protein. Now, I just want to make sure we recall from earlier that a protein is a large macromolecule made up of individual amino acid monomers. So many amino acids bonded together is what makes up a protein. So in this case, so the word translation, I usually think of like English to another language, Spanish or Chinese, right? That's essentially what's happening here because the mRNA is a nucleotide sequence, whereas the protein is made up of an amino acid sequence. So it's like two different languages. So in order to translate between those two different languages, the ribosome has to have a way to make sure the correct amino acid is being added one at a time. And one way that that happens, and we'll go through the process, is based on these three types of RNA. The genetic code is something that is conserved across all organisms, so bacteria, us, plants, and this is essentially the translation between the mRNA sequence and the correct amino acid. We'll look at that in a later video. But I do want to point out that the way translation happens on the ribosome is it reads the mRNA sequence in triplets or in groups of 3. And these triplets are called codons. So I'm just going to put here triplets of the mRNA nucleotides. Now let's get into a little more detail about the nucleotide sequence. As a review, this is a DNA nucleotide over here. And you'll recall that it has 3 components. It has a phosphate, it has a sugar or a deoxyribose, and then it has a base. These 2 components, the sugar and the phosphate, are what make up the backbone, right, or the ribbon of double-stranded DNA. The base portion, in this case, this one happens to be an adenine, is what can be different for each of the 4 nucleotides. So remember that the DNA alphabet, the DNA nucleotide alphabet, has 4 letters: adenine, cytosine, guanine, and thymine. So each of these can be in this position, and then we would have that particular nucleotide base. For RNA, it is very similar, except there's 3 main differences, and the first one that I want to point out to you is that one of the bases is different. In RNA, uracil replaces thymine. So recall that 2 of these are purines, have the double rings. 2 of these are pyrimidines and have the single ring. And a purine is always going to pair with a pyrimidine and vice versa. So for RNA, instead of thymine, we will have uracil. So if we put our RNA nucleotide alphabet, it would be ACGU. Now they pair in a particular way. In DNA, A is going to pair with T and C pairs with G, whereas in RNA, A is going to pair with U and C is going to pair with G. Now I've drawn 2 lines between the A and T and 3 lines between the C and G because that's how many hydrogen bonds there are that are holding those bases together in double-stranded DNA. So as I mentioned, DNA is a double-stranded helix, or in this case a twisted ladder, right, is what we're seeing. This ribbon or the backbone, remember, is the part of the nucleotides that is the sugar and the phosphate component. The rungs of the ladder that are connecting the two backbones are the individual bases and the way that they are hydrogen bonded together. So if you zoom in and look at hydrogen bonding, we see that here on, say this would be one strand or one ribbon, right, running through the phosphate sugar. That would be one of these ribbon backbones. On this side would be the other ribbon backbone, okay, so this one. And in between is the rungs of the ladder. So we see that we have an AT pair here and a CG pair there. So we know that DNA is double-stranded, okay? Now, RNA is not. RNA is actually single-stranded. So that's the second difference we see between DNA and RNA. Our first difference was that there was a different nucleotide alphabet, right? Uracil replaces thymine. Now we see that RNA is primarily single-stranded while DNA is primarily double-stranded. The third difference is going to be in the type of sugar. So over here, this is an RNA sugar. This one is a DNA sugar. The D in DNA stands for deoxy, which we see right here. D means without, oxy, oxygen. And you'll see that there is a difference in this position where we have an oxygen in RNA, but we just have a hydrogen in the deoxyribose. One other thing I want to point out to you before we move forward is you're going to see direction of DNA strands designated 3' to 5' or 5' to 3'. And the reason that it's numbered that way is in a ring structure, like you see, anywhere that two lines meet together and there's not an atom drawn in, it's assumed that that's a carbon. So there would be a carbon here, a carbon here, a carbon here, a carbon here. Now, if I am trying to designate to you which carbon I'm talking about, in the rules of chemistry, there's an accepted practice that you go clockwise around the ring. So from oxygen, we're going to go clockwise. That's the first carbon, that's the second carbon, third carbon, fourth carbon, and the one coming off would be the fifth carbon. So that's where those designations come from. So if I'm trying to tell you this part of the molecule, starting here and moving this direction, then I'm going to say 5' to 3' because it's going this way. So that's where that 5' to 3' designation comes from. Now, we want to understand the three types of RNA. Messenger RNA we've already mentioned. That's what is carrying the message, right, from the gene. rRNA stands for ribosomal RNA, and as you might guess, it helps to constitute the ribosome along with some protein. And then tRNA stands for transfer RNA, and it is transferring that message from the mRNA into an amino acid sequence. And we're going to look at that up close. It has two very important components. The first is it has something called an anticodon, and it will match up with the codon on the mRNA. So that's one part of the tRNA. The other part of the tRNA that's important is an attachment site for the correct amino acid. So in the figure down here, this is sort of a cartoon representation of what a tRNA molecule is doing. So this would all be happening on the ribosome, but the ribosome, the whole thing is not drawn in, just a little bit of this picture for you to see what's going on. So the first part I want you to see is this is the mRNA. This linear sequence you see here is the mRNA message. And you can see that there are nucleotides, right? G, U, C, C, A, G. The sequence of those nucleotides, remember, was ultimately, or at the very beginning, it was instructed for by the DNA. So there was that template DNA molecule, and it was read by the RNA polymerase, and that's how we got this mRNA molecule. Now, the tRNA, okay, remember it's read in triplets, so we have these three, these three. We're focused on this triplet right here and how it lines up with the tRNA. So in mRNA, this portion would be called the codon, and this portion of the tRNA would be the anticodon. And you see that those are complementary, C with G, A with U. So that's one side of the tRNA. It lines up. On the other side of the tRNA is going to be where the correct amino acid is, so that the appropriate amino acids are brought in one at a time and added to that growing new chain of protein. Now, there is going to be a tRNA molecule for every possible triplet combination for an mRNA codon, so that when it matches up, it brings in the correct amino acid. And in a future video, we're going to talk more about the genetic code and how that is translated on the ribosome.